And thanks, David, for inviting me and also organizing this seminar series. I got to see some things around from the science of intelligence and other things that are going on here already. So yeah, it's nice to visit. And excited to tell you more about some work I'm involved in, and also talk more about modeling collective behavior, which is the topic of this course, a topic I like. So to start off, the overview, David already mentioned some different systems I've worked on, which is now quite a few different systems, collective behavior. First projects were with desert harvester ants, now working a lot with honeybees. Also work on different species of fish. Today I'll talk about work mostly with guppies. I've also worked with sticklebacks, golden shiners, and zebrafish. And I'll talk later about C. elegans. I also have some previous and also ongoing work with rodents. So this is a lot of different collective behavior systems. But from a modeling perspective, there's lots of things similar, and also from a data analysis standpoint, a lot of things similar. Some things that have in common of all the things that I'm working on are big data, so to say, that is using quantitative driven analyses and lots of data in order to describe collective behavior and to look at the differences between individuals. The use of modeling to ask questions and to understand the function of the system, and an interest in individual differences, characterizing the system from honeybees, describing different motion and task patterns, to looking at collective motion based on speed differences or social interaction differences. So today what I'm going to talk about, overall outline, I'm going to start off with a kind of more general introduction to modeling collective behavior, more specifically collective motion, and how I think about this, and then introduce a bit more in detail some concepts that I'll then use and show in the two research projects, which one is looking at parasitism and social interactions in groups of guppies, and the other is looking at collective behavior in groups of C. elegans, nematode worms. And then I guess we have enough time for also discussion, so if anything's not clear during, you can ask questions, or of course we'll have time for questions discussion later too. So first, modeling collective behavior and some general concepts. Talking about modeling, I like to start with this slide to say what we're talking about and referring to. The key elements, what I'd say key elements of a model. One is abstraction. This is saying, how do we kind of idealize the system to represent it? So I have a fish, but I can represent the fish as a particle. This is an abstraction, but this allows us to then simplify it into a useful way that's more tractable. Also making assumptions. So I could assume that the fish only interacts with others within a certain distance. And then formalizing of relationships. I like this quote by Richard Feynman, which says this about using mathematics to make precise statements and follow their consequences to a logical conclusion. So to me, this reflects a lot of how models are used, especially asking biological questions, to ask, oh, what if this? What if we change this parameter? What if this were the case? And to understand how this would impact the system and then compare with data. So in modeling collective behavior, and this is more specific to collective motion, I think about different scopes of models and would divide it as so. We could have descriptive models answering the question what. For example, a question can be, what are the interactions between individuals? Another category can be mechanistic models. For example, asking the question, how do individual interactions produce group behavior? And a third model category is a functional, asking the question why, or getting at more like ultimate questions, going from a biology perspective, asking For example, why would certain interaction rules evolve? Or what would the fitness benefit be to having certain? So to start off, I'll give an overview of, I'll give some examples of what would be these different questions. So for a descriptive model, this is an example asking, what are the interaction forces between fish? Here in this work, it represented, okay, first I call this is called a model because this has abstraction. We represent extraction and also assumptions. We're extracting the fish as a point particle and assuming they interact by physical forces. So you can, of course, question this assumption because the fish are not physical particles and they may be doing other things. But making this allows us to precisely quantify how the two fish move in relation to each other. So what this work did is then say, how does this fish respond based on the position and the orientation of this other fish? And based on this, we can make a density map and also then say, how did the focal fish speed up or slow down depending on the position of the other fish, and how do they turn? Basically, they sped up, the other fish is too far away, slowed down if the other fish is a bit farther behind, and turns towards the other fish. Some other follow-up work has used similar concepts, generalizing this to use deep attention networks to look at more different aspects of behavior and also in a swarm setting with multiple individuals. And other work from the Romanchuk group here, looking at force maps based on focusing on relative velocities and the influence of speed for how individuals interact. Another example from my own work, making a seemingly more complicated model, but which actually makes a simplification of decomposing components of distance and orientation to make a more tractable form, is we use this model fit to interactions of zebrafish. So I won't go into the details, but just say how we used it. We use this to ask then, how do zebrafish with different genetic mutations respond to their neighbors? So this model fit then changes in speed based on, and changes in speed and turning based on the neighbor distance and the angles of the others. And by fitting this, then we could have some more precise predictions of how zebrafish with different genetic mutations and responded to their neighbors. So going on to the other category of models, mechanistic models. An example question of this can be, how do individual interactions produce group behavior? So a descriptive model is asking what types of interactions are between individual. But now if we want to ask, how do these come together to produce some pattern? An example of this model, which is used often, is the zonal model, or also called the cousin model, which represents repulsion, alignment, and attraction zones for how individuals interact. And yes, of course, this is a big simplification and making assumptions of how they interact, but with these simple rules then can reproduce different group states and also interesting properties such as hysteresis, and represent a lot of the aspects of motion for going from simple interactions to group behavior. So considering this model, my background is actually physics and then engineering. So I like to think of motion, especially collective motion, starting with a physics birth-based framework. So the framework that I use is based on the Langevin equation, which you've come across this already in other lectures or parts of the course. Perhaps, who's familiar with stochastic differential equations? Okay, good, about half. So this represents, we have a noise term, and then these are changes in speed, or changes in turning rate, or angular velocity. And this represents some drift, actually going back to zero, noise. So this model was called the persistent turning Walker model. It represents a correlated random walk. And then was later extended. The first version included just turning rate, and it was later extended to include changes in speed and other parameters. So we can simulate this, and this was made to more represent fish. And just with these equations, we can also already have something that looks reasonable for representing the motion of, say, individual fish. So combining these frameworks, this is the framework that I've used in multiple models, is using the zonal model rules, but in a continuous time stochastic differential framework, stochastic differential equation framework based on this persistent turning Walker. And an advantage of using this over the original formulation in discrete time is then now we have, well, differential equations which can be well characterized, and you can also add in other forces. And now we have the zonal model rules which come in here as an effective social torque. So the zonal model rules are described as if there is an individual in the repulsion zone, then follow this rule, which means turn away from the average position of individuals in the repulsion zone. Or if there's individuals there's none in the repulsion zone but in the social zone, then follow this rule, which is represents alignment, go towards the average direction of neighbors in this zone, and attraction, the average position of neighbors in this zone. Here's an example of just a simulation of this model. But I will use this actually in both of the projects, the research project I talk about after this. So an example where we used this model to answer research questions is work with Yola-Yolis with schistocephalus parasites. So these, this is schistocephalus. We got these out of the Bodensee. Sticklebacks. Schistocephalus is the parasite. These parasites are really prevalent, and they're also huge. They can be half of the body weight, as you see there in the picture. And we wanted to see how some earlier work with these has suggested that the schistocephalus takes over the nervous system of the animal before it reaches its ultimate host, which is avian, or a bird. So what Yolis was interested in is this actually true, or is it actually more just passive mechanisms? Because if you have a huge parasite, you probably can't swim as well. So some of the observations are that in pairs of fish experiments, the parasitized fish are more likely to be the follower behind the other fish. We wanted to ask, is this due to changes in social interactions, or can this explained by simply changes in speed or swimming ability? This is what a simulation looks like, actually using this model I showed on the last slide. But now we were able to vary the key parameters. That's the strength of social interactions and also the speed difference in the average speed between the two individuals. And doing this in a range of parameters, this suggests that speed has a much stronger effect, and therefore the trends we see in the experiment are more easily explained simply by the passive mechanism of differences in speed. So going on, another example question, thinking of functional models, an example question would be, what are the fitness consequences of social interactions? An example study looking at this question is then asking, how does finding resources, can having interactions, social interactions, improve the ability to find certain resources? And is there an optimal social interactions? This study looked at this and used evolutionary dynamics, looking at populations of one or the other to either have social interactions or not, and looked at social and asocial populations and found that there was indeed an increased fitness benefit to having social interactions, because then we're quicker to find resources. We can go further on this because there are cases where this is not always true, or you don't want to have too high of social interactions. But this is just an example showing the type of question. So that's the overview part. I talked about kind of different types of models and the different types of questions we would ask with these different types of models. And then the things I'm showing next, I'm really going to draw a lot on these two types of models, both descriptive and mechanistic models, in the next two research projects. So now moving on, and now I'll talk about a project looking at parasitism and social interactions in guppies. So now switching gears, I'll motivate this project a little. First, I didn't motivate collective behavior at all starting the presentation, since I assumed everyone is familiar with collective behavior as the seminar is modeling collective behavior. But I will motivate some about this study with guppies and why we're interested in this. So living in a group can be beneficial, as showed in the last example, due to search tendencies, or for example, spreading information about predation threats, mechanisms of collective intelligence, which is a focus question here. But there can also be disadvantages. For example, competition for resources if you're in part of a group, or disease spread. So because of this, different species have evolved different mechanisms to combat disease. Some of these were reviewed in this review paper here. For example, with ants, it's seen that when some individuals are infected, then others change their interactions. So then effectively the interaction network of the ants in the colony change when there are pathogen exposed individuals in there. This could be a mechanism to protect from spreading too much. This is a picture, of course, of social distancing during the pandemic. And here's a picture of a guppy. So guppies are also thought to have mechanisms to limit or spread of parasites. The ectoparasite Gyrodactylus is very common in guppies. It lives on the skin of the guppy, and they can be there can be a lot of them. Guppies are common pets. I don't know if anyone has guppies as pets, but you can get them at pet stores. They're colorful and nice. And if you get them at a pet store, they probably have this parasite. For the experiments we did, actually, Angela got some guppies from the pet store and then took the parasites off of these guppies. So it's very common, that this parasite. Previous studies have suggested avoidance of infected fish. A previous study by Croft et al. Saw shoal fission triggered by an infected fish, but using manual observations and with limited counts of how the fish are swimming together. Other studies by the Stevenson group have used behavioral assays, putting fish in arenas with one side or the other, and have suggested that both visual and chemical cues could be used, but also there's a complex relationship between infection status and also the sex of the fish and how they perceive and interact with the other. So to ask how this parasitism affects the motion in a more natural setting and during schooling, we were interested in the questions, how does parasitism affect collective behavior in the group setting, and what are the mechanisms, how individuals interact, and how does parasitism affecting these mechanisms? This work was done in collaboration with Angela Albee, who did most of the experiments, and also with Ian Cousin at the Max Planck Institute. So what I'll talk about of this is first how I did the experiments and the overview, and then more detail about the isolation mechanisms and differences we see, looking at interaction forces, and then a simulation model that incorporates these findings to look at different mechanisms. So these were collective behavior experiments with guppies. We had multiple tanks, camera above, filming the guppies in the tank. The structure of the experiments was you had baseline and treatment conditions. And in each, there's one focal fish, which we designated with an elastomer tag to ensure consistent identification across trials. The other fish referred to as non-focal fish because they didn't have elastomer tags. Following the baseline tests, then we performed experimental infection, or for the control group, sham infection. And then after some amount of time, retested the fish. Where now we have a focal, possibly infected individual in the treatment condition and other non-infected individuals. So we know the identity of the focal individual, but within the trial, when we can maintain the identity of the other individuals in the trial, also the focal individual, using automated tracking techniques. So this tool is T-Rex, which is developed by Tristan Walter, and this enables, to follow individuals and keep one individual away from the other during a trial. Technically, how this works is it finds instances where the fish are far enough apart and then builds an individual-based model that recognizes one individual versus another. If there's markings or colorations, then the machine learning model would use this. For the guppies, it's mostly, because we're filming with infrared light, it's mostly just differences on size. And then after a few changing the parameters and a bit of manual correction, this works quite well. We manually verified this on some trials, but then we can know that when we look at one individual, we have the same individual identity tracked throughout the trial. So this gives us motion, and you saw some movement of the motion of the guppies, but guppies move a bit complex. They're not always together. Sometimes they're breaking apart and coming back together. Overall, it's called fission-fusion motion. So then we wondered how can we quantify this, and particularly to relate to then parasitism and other aspects. So to quantify this behavior, we came up with an analysis framework based on fission-fusion groups and a distance threshold. So how this works is pretty simple to describe. We say two fish are connected together if they're within a distance threshold, and then fish that form a connected component are called a group. And then we can define groups and events. Groups are connected components, and events are when a group membership stays the same. So with this, we can use this to describe this fission-fusion, how big are the groups, who's swimming with who, and the properties of those groups. So showing, this is with some simulated data, example of what this actually look at. This is using, just some simulated parameters. and some distance threshold. So then when they come close together, then they're connected and counted as part of the same group. And then when we go away, then different group. We can have different group sizes coexisting at the same time. Or also we can have individuals not in a group, simply as one, and called isolated. So overall, when we apply this to the guppy data, we see that individuals spend time in different size groups within a trial. In particular, they're not always swimming all close together. This would be all individuals together in one group. They're sometimes doing this, but usually they're swimming in smaller groups, and a lot of times swimming alone. So now we have a quantitative definition of what is isolated, and this is a group size of 1. So this plot here shows all of the fish, both focal and non-focal, that is parasitized and non-parasitized, in the baseline and treatment conditions. So if we just put all the fish together, then we don't see differences in these distributions from baseline to treatment. So let's look more specifically just at the infected fish. Now, if we look at the infected focal fish, we can also compare the control and the treatment cases and ask, did this time isolated change from baseline to treatment? So first, looking at the control group, we saw that there is consistency, that is consistent individual differences in fraction of time isolated from the baseline to treatment. You can quantify this with a correlation coefficient, and it shows that these differences are consistent. After the parasitide treatment, we saw an increase in the average time that fish spend isolated, and also correspondingly less individual consistency, suggesting that introducing the parasitism is indeed driving changes in fraction of time isolated. So fraction of time isolated is one metric. We can also look at other quantities. We used the average event duration, that's the time spent isolated, between joining other groups, the nearest neighbor distance, and the time in the periphery of group, in addition to speed and turning rate to characterize individual behavior. And with this, we can compare the focal fish to the non-focal fish and the baseline and treatment cases. In the baseline condition, we don't see any differences between the focal and non-focal fish, but in the treatment condition, we see increased fraction time isolated, as well as isolation duration, and nearest neighbor distance, and fraction of time in the periphery of the group, which is defined as being the farthest individual away from the group centroid when swimming with others. Okay, so now we saw these differences, and then wanted to ask, what's driving this? What mechanisms are driving these changes, not only in isolation, but in the other patterns of the data we see? So in thinking about this, drew on this review paper, which I also already brought up in the motivation slides by Stockmeyer, which organized different ways that it could find of different animals deal with disease or parasitism. The most relative important for us, okay, these ones on the left are driven by healthy conspecifics or others. These on the right are driven by the individual themselves, the potentially infective. The ones that are most applicable, we're interested in, are avoidance, that would be changing of social interactions to avoid an infected individual, or either active or passive self-isolation. Passive self-isolation could refer to, for example, what I showed in this with sticklebacks. Then you have a parasite, you're not doing so well, you swim a bit slower, and because you swim slower, then you're more isolated. Active self-isolation would be that you have a parasite and you then interact less socially with others. That is a change in how they interact. So we wanted to ask, do we see these mechanisms in the data, and which ones or which multiple ones best match the trends we see? First, we saw that passive self-isolation does not match trends. This was kind of surprising at first. This would have been the easiest explanation, but we actually don't see differences in average speed or turning motion of the parasitized fish. And other tests with swim tunnel respirometry also showed similar results. There are not large differences in swimming ability associated with parasitism. However, speed definitely influences time and isolation. Other work has shown the effects of speed on collective behavior. We also see this definitely in the data, that there's a negative correlation between speed and fraction of time isolated. That is, slower fish are more likely to be more isolated. And we see these trends both in the baseline and the treatment condition, but in the treatment condition, this trend is offset. So the differences we see between the parasitized focal fish and the other fish are not explained by speed. So next, we thought to take a bit of a step back to have an overall view. As I showed before, tested fish differed in time isolated, particularly parasitized fish are more isolated than other fish. But looking at this data from another perspective, instead of looking at the probability density, now looking at the counts, we see that we have more non-parasitized fish than parasitized fish in the data. And this range of fraction of time isolated fits within the general range of the distribution or the variation observed in the whole population of tested fish. So then we asked more generally, how does isolation relate to all these behavioral metrics and the other things we're measuring in the data? To look at this, we looked at fraction of time isolated in respect to these other metrics, now looking at all of the fish tested in both the baseline and the treatment condition. And now here, 1 point is one fish tested in one trial. And so this shows then there's correlations between some of the metrics and not correlations with turning here. This is turning. And with this, we can say that this allowed us to say that isolated parasitized fish follow the same trends as other isolated non-parasitized fish. That is, we can phrase this a different way, is we can call the correlation between isolation and these other metrics, we're calling this now the isolation behavioral phenotype, for how isolation relates to other metrics. And we can say that parasitism increases the likelihood of the isolation behavioral phenotype. And now with this view, now we have data from all the fish. So now we really have a lot of data and we can compare these interactions in detail. So what we did next is calculated effective pairwise forces. So really using almost, going back to what I showed first, a type of descriptive model representing forces and the interaction between individuals. Here, making the simplification that the fish we're tabulating forces with nearest neighbors because we have more than two fish swimming together. And to now use all the data, we made a simple quantile division and compared less isolated fish with more isolated fish, dividing at the 0.75 quantile for how these fish interacted with others. Then we could say, how did less ISO fish respond to their nearest neighbors, and compare this to how more isolated fish respond to their nearest neighbors. And now looking at the fish in these two categories, we can calculate the effective speed and turning forces here. So what this is actually looking at, this is the position. Here we have a fish at the center, which is either a less isolated category or more isolated. This is facing this way. And then here's the position of the nearest neighbor, X and Y. And this is how the fish turned in the other direction. We take an average here, and this is the same thing done for speed. Now this is changes in speed. So if we're looking at within this line, then is an average turning response plotted here, and within this line, the speeding is mostly front and back, and turning is mostly left and right. We can project these onto a one-dimensional simplification here. And so this shows that, okay, this more isolated fish, this is about the same, but this is lower. This is less magnitude. So this represent when there's another fish a bit farther away, they're not turning as strongly towards that fish as the less isolated fish would be. So this is indeed a mechanism that would drive more isolation. If your nearest neighbor is a bit farther away and you're not turning very strongly towards them, then you may lose them and become isolated. The speed response, it's kind of hard to see a clear difference here. But the turning response seems definitely of something that fits within this self-isolation by turning less towards others far away. So next, we used a similar framework to ask about avoidance mechanisms. So with this, we have we put a less isolated individual at the middle, and then said, what is the conditional response if the neighbor is either another less isolated individual or a more isolated individual? So this is then the conditional response based on the category of the neighbor. We calculated the same thing. Now, here the turning response, we see no difference, but the speed response, we see a difference that seems more clear. So what this is representing here is when there's a less isolated individual at the center, a more isolated individual is the nearest neighbor. This effectively means, here it would be speeding up or slowing down to keep pace with the other, and these zones are essentially elongated. So the speeding response is in a way that they're not speeding up as much to keep pace with these neighbors, and they're not slowing down as much so they don't lose them. And this is indeed a mechanism that would drive avoidance. But different from the isolation mechanism, this is speed-based, not turning-based. So we don't see differences in the speed responses. But overall, from the data, the results just suggest both mechanisms can play a role in this. And key is that speed modulation is also important. So now, then taking this, doing the same calculations for just the focal and non-focal fish in the treatment condition, we have a lot less data because there's less fish. Generally, we see the same type of mechanisms, but it's a bit noisier because we're using less data. That is here, some difference in the turning response. Here, a bit more repulsion instead of less attraction, but still in the same conceptual mechanism of difference in turning response that would promote self-isolation, and a difference in this speed response. So next, with this data, we then wanted to incorporate this into a model to ask, okay, if we have these different mechanisms, how would this predict other aspects of the metrics and what we see in the data. We use this modeling framework that I previously showed, the zonal model in the persistent turning Walker modeling framework. And we added a speed rule to then adjust speed for the neighbors. And to represent these different mechanisms, we represented self-isolation by the agent has a lower social weight than others around, and avoidance by there's a less weighting to some certain individuals. So this is so-called selective interactions based on the identity of certain individuals. And mathematically, this was represented by introducing a coefficient or coefficient matrix beta, so that when your neighbor has a certain identity, then you have a less weight put to this neighbor when making the averages for the attraction and alignment in the zonal model. Briefly on the methods, how we did this is then set certain parameters based on previous work and comparing with the data, and then tuned the key parameters related to each mechanism by fitting the distributions of time isolated. And with this, we simulated a parameter grid, and then with this, then are able to then use a fit of these distributions, and then with these fit parameters for self-isolation, that's the difference in the social interaction for weight versus non-focal fish. For the avoidance mechanism, this is the social interaction weight of all, and then this selective interaction parameter, and then by setting these, we can then simulate to see how this compares with other metrics we see. So overall, we see that both mechanisms produce the core experimental trends. And particularly, they can both fit differences in fraction of time isolated from parasitized to non-parasitized fish, and also differences in nearest neighbor distance and periphery of the group. However, the speed-based avoidance mechanism provides a bit closer match, predicting more of a distance in nearest neighbor distance with this model, and also not a difference in turning rate. We don't see a difference in turning rate in the data, and the self-isolation mechanism simulated gives a large difference in turning rate because of the different rules. And now we can plot this in different ways related to the data. Here actually the dashed line is the data, and the points are different agents from the simulation. Because I guess I failed to mention, because of the key role of speed, then we performed multiple simulations and drawing the speed of the individuals from a Gaussian distribution for each simulation. So that's why we have many different points, and we run many different simulations to get a distribution because we're drawing speed for each case. So overall, we can see, we can reproduce most of the isolation behavioral phenotype with both of these mechanisms. The avoidance-based mechanism provides a bit of a closer match. So summarizing this part is we don't see overall differences in motion, but we see differences in isolation. And using this force-based methods as well as the simulation suggests that both mechanisms of self-isolation and avoidance can play a role, with the avoidance mechanism providing a bit closer match to the trends we see in the data. So now I'm going to switch topics to talk about another study, which is also going to use a similar modeling framework. And this is current work looking at collective behavior in heterogeneous groups of C. elegans nematode worms. The motivation for this work, as previous work shows how the genetics relates to behavioral differences, particularly in collective behavior and aggregation differences. We showed NPR, by changing a single gene, we can have worms that don't aggregate versus that they do aggregate, and this we can manipulate this. However, if we would find worms in the wild, then they're not they're rarely uniform groups. But we were wondering how is the behavior of the group shaped by the mixture? What if we put worms with different behavioral traits together? How does the collective behavior come out? So this is work with Serena Ding and Narcisse Font, who are doing the experiments. And overall, what the experiments we did is we have collective behavior experiments with 40 worms using fluorescent markers on the head to distinguish the different worms. And this looks like, so you can see fluorescent markers of the worms in a dish. And we performed homogeneous trials with all of one type for 3 different genetic lines, N2, which tends to not aggregate, NPR1, which is known to aggregate, and CB, which is a Hawaiian strain, which also seems to aggregate. We performed homogeneous trials and then 2 different cases of mixed trials, putting N2 and NPR1, I'll call these A and B for shortness, and then putting B and C together. Look at the properties of these mixtures. So first, quantifying the aggregation behavior. To quantify this, we use the pair correlation function, which is also called the radial distribution function, which is the probability of finding another individual at a certain distance. So if this is 1, this is a random distribution. From this, we can compare, this is the homogeneous cases of cases A, B, B and C. See that indeed, A and 2, this is lower than B, showing the differences in aggregation behavior. As well, both B and C are both aggregating, but NPR strain B is aggregating more. And then this middle line is the heterogeneous case. So if we calculate this on all worms, in each case, then the line falls between the 2 homogeneous cases. To look at more specifically, then we calculated the strain-specific pair correlation function. So that is just type A with type A, type B with type B, and type A with type C. So this shows, kind of surprisingly, this curve with just here B with B in the heterogeneous experiments looks similar to that in the homogeneous experiments, and similar trends for the other ones. So this can be explained if there's some segregation between the different types. To quantify this, we use the segregation parameter, which calculates density overlap. And it shows that in the first mixture, which is the A and B, we saw more separation than in the second mixture. So next we looked at movement of the different strains. So this is speed, and this is the homogeneous cases and the heterogeneous cases. Their case strains A, B, and C, but we see that overall B moves a bit faster than A or C, and that from the homogeneous to heterogeneous cases, there's not a significant change in the average speeds. Other work has shown that speed changes with density are needed for swarming. So we looked first to see, do we see this? And also ask, does this depend on the identity of the neighbor? So this is, we define local density as the number of worms within a certain area around a focal individual, then asked, how does this speed change with local density? And so strain A, N2, does not change. And for both B and C, this does change, with it changing stronger for strain B. And key here, then the shape of this curve does not depend on the identity of the neighbor or the composition of which worms of which different strain are close by. So to further ask how these mixtures come together with these different properties, and can we predict the mixture properties, we used a simulation model adapted to represent worm motion. So previous work has looked at detailed Brownian motion-based model with added reversals to represent nematode behavior. That was done in this paper. So what we did is then combine this quantitative framework with the zonal model for interactions. So now we have worms move by Brownian motion, because worms don't have momentum like a fish. It's more Brownian motion-like. And then added interactions based on the zonal model. So this comes in actually the same way as in the case with fish. This preferred direction is set by the zonal model. And to simulate, first we matched the speed based on local density to the data. And then, similar to the work with the guppies, we simulated a range of parameter values, here just varying the social interaction weight, which is the weight of turning towards this preferred direction, into the data. So by changing this, we can change aggregation behavior as quantified by the pair correlation function. And then we can match this for the different cases to the data. And then we simulated this with different mechanisms. That is, worms interact with all others or only interact with the same type. So this shows, so this is still a work in progress. First though, we show that the model can match the different cases for matching the simulation to the experiments. We're working on refining this. It can actually fit a bit closer to what's shown here. And then simulating with all-to-all interactions generally matches the trends in the data, looking at the heterogeneous cases. That is, we have, we can look at the strain-specific pair correlation functions, and we see similar types of trends. Then we were wondering, what if we have selective interactions and essentially ignoring the other type of strain? And the key here, now we have this cross function for interaction with the other type, which goes to 1, which means random interactions. And this is not what we see in the data. We see this actually more between or close, what's shown on the left. And you might be wondering, why are we interested in selective interactions, or why would we think of this? Actually, in other nematodes, it's been shown recently that there are selective interactions related to kin recognition that are controlled by a different genetic pathway and can be manipulated. So this was shown in P. pacificus. And so we have an adult worm and some larvae, and they cannibalize larvae of non-kin, but they avoid eating kin. And it's thought that this is interesting because this is a very simple organism and a single pathway that can be manipulated associated with its behavior. So it's interesting that this is a tractable system that could ask, how is this kin recognition implemented, and how did this evolve? So in current ongoing experiments with P. pacificus, we're looking at closely related and more distantly related strains, and then using this quantitative analysis framework and simulations to then understand this behavior and how they mix together. So overall, summarizing this part, we saw that strains differ in mobility and aggregation behavior, and that for C. elegans, interactions are non-selective, and by fitting parameters to a homogeneous case, we can predict the cases of the mixture experiments. And ongoing experiments are focusing on selective interactions in P. pacificus. So now, overall, kind of summarizing, I talked about a lot of stuff, all modeling and collective behavior related. I first talked about overall modeling concepts, different types of questions, and different types of uses for models. Then I talked about social interactions and isolation in guppies, and then collective behavior in heterogeneous groups. Before I conclude, I just want to briefly mention some other projects I'm involved with. In case someone interested, happy to chat. My main experiments now which are at FU Berlin, is looking at the effects of pesticides in honeybee colonies. Also currently working with a group Martha Havenig's group at Ernst Strongman, looking at the effects of psilocybin on social behavior in rodents. And with our collaborator Ahmed El-Hadi, we're also working on a startup company, Ecodilic Science, and we have funding through Maximize in order to test out these ideas. So be it applications of behavioral analysis in applied fields. So with this, I want to thank others, acknowledge those involved, and especially the work I showed, Angela Albee, who did most of the experiments with guppies, and Serena Ding and Narcisse Font, who did the experiments with C. elegans. And yeah, thanks for having me here and listening too. And that's it.